Hello students, welcome to Misostudy. So we will be discussing about some questions which are important for the chapter Biotechnology and Application in respect of your NEET exam. I have collected some 10 questions which are important for your NEET exam, and from that biotechnology applications, in the previous chapter we have studied about the biotechnology processes. In that we have a clear-cut idea that how this restriction enzymes they are used, why they are important. Similarly, the vectors are also important. In this whole chapter biotechnology application, we have studied some of the examples by which examples are some hormones which we have produced with the help of restriction enzymes, vectors, etc. And at the end, what we want is a desired product, and that we have made with the help of fermenters, right. Hope the whole procedure is clear to you. In this whole chapter, we have talked about some important procedures, we have talked about some important examples that we have already prepared. For example, the BT cotton, BT brinjal, etc. we have prepared. We have prepared the human insulin also with the help of biotechnological processes. Similarly, we have prepared the growth hormones also with the use of the processes that we know till now. So let's talk about the 10 questions. I'll discuss all those questions one by one. Not only the one option, we'll discuss the rest of the options as well, right. And I have collected in and out over some questions from the previous years, because as per their frequency in the previous years, the questions frequency in the previous years, this shows that the following topic which I have included over here, they are important for your NEET exam. Mostly they used to ask such questions like this only, and the topic remain the same. So let's start the first question. The first question indicates that the two polypeptides of the human insulin are linked together by. I have already discussed the insulin is prepared, this recombinant insulin is prepared in the E. coli. When we see that there are two chains, the A chain and the B chain, similarly in the proinsulin form, the C chain is also present. Let's look at the structure of these. Let's look at the structure. We have two chains, one is called as a A chain and second is called as a B chain. B chain, like this. And when we see that in case of a normal or a proinsulin, as it is produced in the proinsulin form, a C chain is present. What is this? This is a C chain. What is this? This is a B chain, or B peptide or C peptide, whatever you want, you can give that name. Similarly, we have A chain like this. What is this? This is a proinsulin. Proinsulin. This proinsulin is converted into the insulin form. In the post-translational modification, this particular protein is converted into an active form, and that active form is called as insulin, and this is called as a post-translational modification. Post-translation modification, and with the help of this particular mechanisms, what we have is the two chain, one is called as a chain and second is called as a B chain. A chain and second we have is a B chain. Now, the characteristic feature of these two chains is that they are connected to each other, and the bond which is present in them is a SS bond, that is also called as a disulfide bond. Not only this is present within the two chains, in the two chains, this is also present within the chain as well. So this is an active form which is called as a insulin. Now the question indicates that which type of bond is present in the insulin, that is called as, this bond is called as disulfide bond. So the correct answer for the question number 1 is a disulfide bridges. Come at this side, the option C is the correct option, right? Now let's discuss the next question. They are discussing in vitro clonal propagation in plants. In vitro clonal propagation is characterized by, in clonal propagation, that means what we have to do, we have to prepare a clone of it, and we know that we have a very clear cut mechanism we have studied in the biotechnology principle and processes, that is called as a PCR. And what is the full form of PCR? PCR full form is polymerase chain reaction. You must be worried why they have written RAPD here as well. RAPD means randomly amplified polymorphic DNA. There is a characteristic difference between PCR and the RAPD is that in case of the polymerase chain reaction, they produce a copy of a specific gene. Whereas in case of the RAPD, randomly amplified polymorphic DNA, they produce random genes, they amplify random genes. So, this is a characteristic feature, that means they produce copy of specific gene. Whereas these produce copy of random segment. So, this is the difference between RAPD and PCR. So, the correct option is option number A. We will not stop here, we will look for the other option as well. The B option indicates northern blotting. You must have heard of western blotting. You must have heard about the western blotting, etc. So, let us talk about western blotting. Southern blotting. And northern blotting. Northern blotting. There is a difference between all these type of blotting. As the name indicates, let us talk about western blotting first. Western blotting, that means we are separating fragments of DNA. With the help of electrophoresis, we are separating the segments of DNA. That means it is used for DNA extraction, DNA not extraction, for the separation of DNA fragments. Next we have is a southern blotting. Sorry, this western blotting is used for protein, basically purification of proteins, whereas this southern blotting is for DNA and this northern blotting is for RNA. So, this you have to remember. Western blotting is for proteins, for the protein purification. In this case, the SDS page is used. SDS page, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. The protein separation is done by this method. Southern blotting, that is used for the separation of the DNA. The fragments of the DNA is separated by southern. Similarly, for the RNA, mRNA, we use northern blotting. So, this you have to remember. So, the correct option is A, that means the B is an incorrect one because it is related to the RNA. Let us talk about the C, electrophoresis and HPLC. You know about the electrophoresis, that means we are separating DNA. As we are separating DNA, that is called as electrophoresis with the passage of electricity. This actually happens. Next, it is written over here HPLC. This term might be new for you. HPLC is high pressure liquid chromatography. The name itself indicates that means a chromatography is occurring. We are separating solute as well as solvent. So, this is related to that. It is a quite efficient technique for the separation of the solutes from the solvents with the help of solvents. So, this is not related to the in vitro clonal propagation. Next we have is a microscopy. We all are acquainted with the microscopy. You must have used this microscope in your schools. So, hope you are clear with the microscope. Microscope means we are preparing an image and that image is much larger as compared to the original that specimen. So, that this could be easily visible to our eyes. So, this is related to that. So, the correct option for the question number 2 is option 1, that is PCR and RAPD, right. Let us talk about the next question that is related to the cry gene. Cry gene. Cry gene is obtained from Agrobacterium tumefaciens, Bacillus thuringiensis, Rhizobium leguminosaccharum, Rhizobium fascioli. So, this is written over here. Now, we have to look for the cry gene. Hope you remember when I was teaching you about the BT cotton, how this BT cotton is produced. Now, let us understand this. BT cotton is produced by incorporation of one gene and that gene is called as cry gene. Cry gene produces certain toxin, some endotoxin they are produced, and those endotoxin when they are engulfed by any of the insect, that endotoxin it is in a pro form. Later on with the help of alkaline nature which is there in their gut, it is converted into the active form and this causes a death of that particular insect, right. With the help of recombinant DNA technology, with the help of recombinant DNA tools, what we have today is that particular mechanism by which we have inserted this gene, that cry gene into any of the plant, and such as a BT cotton we have, a BT brinjal we have, BT potato we have, that is inserted into that particular plant. So, whenever an insect will come over there and they will try to attack such plants. In that case, what will happen? Those cry gene which is there, they produce certain toxin that crystallize, that certain crystals they will produce, and that crystal when they are engulfed by that insect, what that insect that will be converted into an active form and they cause the death. Basically, various Coleoptera, Lepidopteras are there that is used, or basically they are used to prevent such type of infection. And let us talk about the cry gene. Cry gene, this is produced by Bacillus thuringiensis, produced by Bacillus thuringiensis, and this particular gene impart insecticidal property. This cry gene produce what? Endotoxin. They produces one endotoxin that is in the form of a crystal, right. So, this you have to remember, that means that they are produced by the Bacillus thuringiensis. Let us look for the option. The cry gene is obtained from Bacillus thuringiensis, the option B is correct. Now, how you can, if you do not remember this cry gene, how you can relate to this? Agrobacterium tumefaciens, this help in the transformation, and basically they causes crown gall disease. It's like a tumor condition. They produces a tumor condition, and what they do, they insert their DNA, and with the help of using non-pathogenic form of Agrobacterium tumefaciens, we can produce. They can help us in a transformation, that is, a transferring our desired gene. Let's talk about the C. That is C and D. They both are related to the rhizobium. Rhizobium, they remain associated with the roots of the leguminous, and they help in fixing of the nitrogen. So the exact option which is left is a B option, and with the help of option elimination you can also answer such question. That is a Bacillus thuringiensis. So this was the question number 3. Correct option is a B. Let's look for the question number 4. This is also related to the Bacillus thuringiensis. As I have told you that they produces cry gene. They have a cry gene with them. Let's look at the question. The inactive prototoxin is inactivated in the gut of insect bile. Now, what we have, as I have told you, the cry gene is there, and that cry gene, that particular gene is taken, and with the help of recombinant DNA technology, is inserted into many of the plants, and the name of that plants, let's talk about the cotton first. Inserted into the cotton, and whenever an insect they will attack, we know that this cry gene, they produces toxin, and that toxin is in pro form. Pro means inactive form. When the insect they will attack, they will try to feed on the cotton plant. This protoxin is converted into active toxin. When it is converted into protoxin from the active toxin, basically they require one factor for their conversion, and that factor is an alkaline pH which is present in their gut. So what we need is a alkaline pH. Right. Here I have discussed about the insect attacked, and those insect they can belong to the order Coleoptera, in the Coleoptera beetles come, or in the order Lepidoptera. In this the beetles come, and in this the Lepidoptera, you can say the butterfly comes, right. So the correct option is the alkaline pH, that is option number B for question number 4. Let's talk about the next question, which is related to insect pest resistant, the same question related to the cry gene, so I am not rubbing this. Insect pest resistant BT cotton plant was developed using. Now come at this side, because what we are doing, we are with the help of recombinant DNA technology, I am taking out this gene and I have inserted this gene into the cotton, and the process which I have used is recombinant DNA technology. Now what has happened? This gene is inserted into the cotton. That means this clear cut indicate the transgenic technology is used. That means we are transferring DNA. Whereas the other somaclonal variation, micropropagation, somatic hybridization, they combine, they are tissue culture methods. Tissue culture method. That means for the question number 5, we need some insertion of the gene. That's why the D option is a correct one. Let's talk about the next question. Strategies to prevent. This is also one of the good question, one of the latest techniques. The strategies used to prevent the nematode infection in the roots of the tobacco plant is. Now they are discussing about one nematode. which affect the roots of the tobacco plant and they causes decrease in the yield, and the name of that nematode is Melidogyne incognita. Melidogyne incognita. Because of this infection, the yield of tobacco is affected. Yield of tobacco affected. Because the yield of tobacco is affected, yield is affected. Now we are focusing on one of the latest technique which is related to the RNA interference. Correct option is RNA interference, iRNA. Now how this iRNA is related to this? When I was talking about this session, the whole chapter sessions, I have already discussed about this RNA interference and why this is important. This Melidogyne incognita, they incorporate their gene that causes the yield is affected. Now how this can be this endotoxin which is produced by one gene, which is, I am talking about the DNA. Endotoxin which is related to Melidogyne incognita, they produce certain kind of gene, endotoxin, and that endotoxin is coded by one DNA. And that endotoxin, what the procedure we are following, in the transcription process, this endotoxin, this gene for the endotoxin, the DNA, they code for one mRNA. Now, with the help of this mRNA has a tendency to produce protein, and the process of producing protein is called as a translation. Now, what does this RNA interference means? RNA interference means I have produced one iRNA, and that iRNA is complementary to this particular mRNA. I have inserted that. When it is complementary to that, now what will happen? This iRNA, they will come and they will bind to this mRNA. When they will bind to this mRNA, the translation will not happen. That endotoxin, which is in the form of the protein, that will not be formed. In total, what will happen? The yield of the tobacco plant will not get affected. As it will not get affected, so the whole procedure is based upon the RNA interference, iRNA. So this you have to remember, because lots and lots of research is going in this particular direction, and this is the most specific mechanism to control the disease in case of any plant. So the correct option for the question number 6 is the RNA interference. Let's talk about the next question. Transgenic crop. It is related to the transgenic crop. 7th question. Transgenic food crop which may help in solving the problem of night blindness in developing countries is. Now tell me one thing, which of the following, just tell me one thing, which vitamin is responsible, whose deficiency of which vitamin is responsible for the problem of night blindness? That is related to vitamin A. Vitamin A deficiency. Vitamin A deficiency lead to night blindness, right? And the name of vitamin is beta carotene. Beta carotene. Now, as I've told you that this is the most advanced form in which we are inserting a gene of interest, so that the disease could be prevented in any of the ways. Golden rice is that rice which has a gene for beta carotene. So whenever we consume this particular golden rice, this vitamin A, the pro form, that is in their pro form, is converted into the active form, so that we can consume this vitamin. And whenever we consume that, the chances that in the life when we develop the chances that within the life the chances of development of night blindness is very, very, very less. So the correct option is the golden rice, right? For the 7th, the option B is correct. Let's talk about the next. It is related to ADA deficiency. Related to ADA deficiency. Now what do you mean by ADA? ADA is adenosine deaminase. Adenosine deaminase, whatever we food consume, in that if adenine is present, that is digested by or that is metabolized by adenosine deaminase. This is one of the function. So, metabolism of adenine. Adenine is what? It is a nucleotide. So, they help in metabolism of adenine. Now, they are talking about the adenine deficiency. Hope you have heard of the one deficiency which is called as SCID. SCID is related to ADA deficiency. In patient, if there is ADA deficiency, that is called as, that particular disease is called as a SCID. That is severe combined immunodeficiency. Severe combined immunodeficiency. In the patients of SCID, the ADA is not formed. ADA not formed. When ADA is not formed, in that case, the ADA is responsible for the formation of WBC. Help in formation of the WBC. So, the WBC will not be formed. When WBC will not be formed, then the whole immune system will be hampered. When immune system will be hampered, now what will happen? Will the patient will be able to respond to any pathogen which enters into their body? No, they will not be able to respond. The pathogen will come, they will attack on the body, they will cause a destruction over there, and ultimately this could lead to the death. The patient can only survive in those conditions if they are kept in sterilized environment. Sterilized environment, then only it will be able, the patient will be able to survive. Otherwise, various treatments are also there, like this enzyme replacement therapy. What we can go for? We can go for enzyme replacement therapy, where we insert this gene for, where we insert this enzyme, that is ADA. We can go for enzyme replacement therapy, which is called as ERT, or we can go for stem cell therapy. These are the two ways by which we can prevent such type of patients. We can insert those gene, or we can replace those stem cells, right? And this is related to the immune system. Now come at this side so that we can look for the option, the correct option. The first indicate increased risk of infertility. Is this related to that? No, not at all. Inability of immune system to function, this is the right option. Next is a chromosomal disorder. No, this is not a chromosomal disorder. Decreases the yield of a crop. This is also very irrelevant because it is not related to plant, it is related to the human beings. So, hope you are clear with the question number 8. The option B is the correct one. Now, let's look for the question number 9. The Bt cotton is resistant to. Bt cotton, we have already studied Bt cotton, that is Bacillus thuringiensis. Bacillus thuringiensis, they produces endotoxin, and those endotoxin, they causes death of the various pests, and basically that pest, they are insects. And for example, Coleoptera, we have discussed that. This is a beetle, the order Coleoptera and order Lepidoptera. I am talking about the class Insecta, and these two which I have written above, they are orders, right? They are resistant to the insects. and the gene which is responsible is a cry gene that we have discussed. Let's talk about the question number 10. Which one of the following bacterium is used for the production of transgenic plants? Now, what are transgenic plants? Transgenic plants, they are those plants which are having a foreign gene into them. When they are having a foreign gene, that means we need a certain vector by which we can transform that particular original cell. So, let us look for the options which are having these vectors. E. coli is a vector? No, E. coli is not a vector, rather than the plasmid which is present in the E. coli, that is a vector. But this E. coli cannot be used in case of the plant because E. coli does not infect the plant. Next is Pseudomonas and Streptococcus aureus. These are wrong option. The D, they have written over here the Agrobacterium tumefaciens. Agrobacterium tumefaciens, like we have discussed earlier also, they causes crown gall disease. Normally they causes crown gall disease. And for the transformation experiment, for inserting a foreign gene, what we do, we produce a non-pathogenic form. And this non-pathogenic form, because we know that they have a capability to insert their DNA, they have a very good transgenic capability. So, what we can do, we take that non-pathogenic form and they help in insertion of gene of interest into a plant cell. And ultimately, when the insertion is complete, what I will get is a recombinant cell. Recombinant cell, right? So, the correct option is the D option, Agrobacterium tumefaciens, right? Hope students you all are clear with the 10 questions which we have studied. These 10 questions, these are important, I would rather say, and most of the questions they were related to Bacillus thuringiensis because the frequency is higher as this Bacillus species, Bacillus is considered. So, this you have to remember. Students, this is all about the today's session. We'll meet in the next session. We'll discuss more questions.